In this section, we'll discuss different components of anticoagulant system. Anticoagulant system prevents formation of blood clot in normal circulation and also prevents spread of blood clot beyond site of vessel injury. In contrast, fibrinolytic system dissolves blood clot that has already formed. Both these systems maintain patency of blood vessels. In this section, we'll only discuss anticoagulant system. For better understanding of anticoagulant system, let us briefly review normal process of blood clotting. Formation of blood clot involves formation of platelet plug initially and then production of fibrin during clotting cascade. Von Willebrand factor and tissue factor are present in subendothelial region of blood vessels and are exposed after endothelial injury. Platelets bind to von Willebrand factor and initiate formation of platelet plug. And factor 7 binds to tissue factor and this starts clotting cascade. Then tissue factor pathway results in activation of factor 10 that results in production of thrombin and then fibrin. Thrombin then further activates platelets and clotting factors of intrinsic pathway. That results in generation of more thrombin to achieve hemostasis. Different components of anticoagulant system then inactivate these activated clotting factors beyond site of clotting reaction. Before we discuss components of anticoagulant system, we know there are two important cofactors in clotting cascade that are factor 5 and factor 8. These cofactors result in significant propagation of clotting cascade. Activated factor 5 with activated factor 10 converts prothrombin to thrombin, and activated factor 8 combined with activated factor 9 results in activation of factor 10. There is a special mechanism that results in inactivation of both these cofactors that we'll discuss later. Now we have reviewed clotting cascade. Let us discuss different components of anticoagulant system. As first step in formation of blood clot is platelet adhesion and aggregation. So in normal circulation, several factors prevent binding of platelets to endothelium and their activation. These factors are prostacyclin, nitric oxide, and ADPase. Similarly, there are three mechanisms that inactivate activated clotting factors. First is tissue factor pathway inhibitor that inhibits tissue factor pathway. Second is antithrombin that inhibits or inactivates thrombin, factor 10, and several other clotting factors. And finally, thrombomodulin with protein C and protein S inactivate factor 5 and factor 8. Along with these specific measures, there are some general factors that also prevent formation of blood clot in normal circulation and also prevent spread of blood clot. First of all, in normal circulation, intact endothelial cells provide barrier to subendothelial procoagulant factors like von Willebrand factor and tissue factor. In addition, all clotting factors are produced in inactive form and these factors are only activated after vessel injury. And when activated, these clotting factors are kept at site of injury because clotting cascade mostly occurs on platelet surface provided by platelet plug that is formed at site of injury. Now let's discuss specific components of antiplatelets and anticoagulant system. And as formation of platelet plug is first step in formation of blood clot, we'll first discuss factors that prevent platelet adhesion and aggregation in normal circulation. In this diagram, endothelial cell is on top and underlying vascular smooth muscle cell below. Prostacyclin is produced from arachidonic acid in endothelial cells that reaches to smooth muscle cells and increases cyclic AMP in smooth muscle cells. And nitric oxide is also produced in endothelial cells by nitric oxide synthase. Nitric oxide also diffuses out and it increases levels of cyclic GMP in smooth muscle cells. Both high levels of cyclic AMP and cyclic GMP decrease calcium in smooth muscle cells, and low calcium levels prevent contraction of smooth muscle cells of vessel wall. This favors vasodilatation and normal blood flow and prevents stasis. Same mechanism prevent platelet adhesion and aggregation as well. In this diagram, platelet is at top and endothelial cell below. Prostacyclin produced in endothelial cell in same way increases cyclic AMP in platelets. And nitric oxide produced in endothelial cells increases amount of cyclic GMP in platelets. These changes ultimately result in low calcium levels in platelets like in smooth muscle cells. And low calcium levels in platelets prevent granule release and prevent activation and aggregation of platelets in normal circulation. However, after vessel injury, thromboxane A2 and other factors result in opposite reaction in platelets, and that results in release of platelet granules and platelet activation. Along with thromboxane A2, ADP is another powerful activator of platelets. To counteract this, endothelial surface have an enzyme called adenosine diphosphatase or ADPase. This enzyme degrades ATP and ADP to AMP, and that prevents activation and aggregation of platelets. So prostacyclin, nitric oxide, and ADPase prevent platelet binding and aggregation to normal endothelium. Now let's discuss different components of anticoagulant system, that is tissue factor pathway inhibitor, antithrombin, and thrombomodulin. Anticoagulant system helps to prevent excessive activation of clotting factors at site of injury and also inactivates activated clotting factors beyond site of injury. We know clotting cascade is initiated by tissue factor, and that pathway is called tissue factor pathway. This pathway is inhibited by tissue factor pathway inhibitor. Tissue factor pathway inhibitor is bound to endothelium and is also present in blood. It inhibits factor 7 tissue factor complex and factor 10, and this prevents tissue factor pathway from further production of thrombin. Other anticoagulant factors inactivate clotting factors that are part of intrinsic pathway, common pathway, and cofactors. One of main anticoagulant factor is antithrombin that inactivates several clotting factors including thrombin and factor 10. Antithrombin is a protein present in blood. When circulating thrombin binds to antithrombin, it results in inactivation of thrombin. Antithrombin also inactivates factor 10 after factor 10 binds to antithrombin. We know thrombin and factor 10 have central role in clotting cascade, and as antithrombin inactivates both these clotting factors, so antithrombin is an important anticoagulant. Antithrombin can also inactivate other clotting factors like factor 9, factor 11 and factor 12. So antithrombin can inactivate most clotting factors, especially of common pathway and intrinsic pathway. Rate at which antithrombin inactivates thrombin and factor 10 is significantly increased after antithrombin binds to heparan sulfate. Heparan sulfates are bound to surface of endothelium. When antithrombin binds to heparan sulfate, it results in conformational changes in antithrombin. These conformational changes significantly increase rate of inactivation of thrombin and factor 10. So heparan sulfate increase rate at which antithrombin inactivate these clotting factors. As antithrombin is important component of anticoagulant system, so deficiency of antithrombin predispose to hypercoagulable state. Another important component of anticoagulant system is thrombomodulin. As name suggests, it modifies actions of thrombin. We know factor 5 and factor 8 are two important cofactors in clotting cascade. Thrombomodulin results in inactivation of these cofactors. This process starts when thrombin binds to thrombomodulin. After thrombin binds to thrombomodulin, it then activates protein C. Activated protein C along with protein S inactivate cofactor 5 and cofactor 8 present in blood. Deficiency of protein C or protein S result in hypercoagulable states. Similarly, if factor 5 has mutation, then activated protein C cannot inactivate factor 5, and this also leads to hypercoagulable state. In summary, we have discussed there are several antiplatelet and anticoagulant factors that prevent formation of blood clot in normal circulation and also limit spread of blood clot beyond site where hemostasis is needed. And many of these antiplatelet and anticoagulant factors are related to endothelium of blood vessels.